Hello and welcome everybody to my channel Omni Bio Entrance, and today we will be discussing Delhi University MSc Entrance Examination for the subject Microbiology conducted in the year 2021. Coming to the first question. The complement cascade has four major biological functions. Which one is not one of those? Membrane disruption and lysis. That is a function. Opsonization, that is also a function. Suppression of inflammation. It is not suppression, it is activation of inflammation. So option 3 is the right answer. Then which one of these is not a professional antigen presenting cell? So professional antigen presenting cells are immune cells that specialize in presenting an antigen to a T cell. So B cell, dendritic cell, macrophages are professional antigen presenting cell, whereas T cell is not a professional antigen presenting cell. Then next one. The major burden of defense against intracellular viruses in innate immunity is shared by. So NK cells, that is natural killer cells, they destroy virus infected cells by inducing the infected cells to undergo apoptosis. Where cells infected with virus also produce interferons, that is type 1 interferon, which activate killing activities of NK cells. So the answer here is type 1 interferon and NK cells. Answer is option 1. Then next one. IgG, IgM are not mediators of which type of hypersensitivity? So it is type 4 hypersensitivity because this is a cell mediated reaction that occurs in response to contact with allergens. So answer is option 2, that is type 4. Then in 1909, Wassermann introduced a test which proved to be sensitive, rapid and did not require any special instrument or expensive chemicals. The test is used for diagnosis of many bacterial and viral diseases including syphilis. Which test is it? So it is called as complement fixation test. Answer is option 4. Which one of these is not a major family of cytokine receptors? So class 1 receptor and hematopoietic are family, class 2 receptors, all these are cytokine receptor families. IgG superfamily is also a cytokine receptor family, whereas LTE superfamily is not. Answer is option 3. Then what type of DNA has glycosyl bond conformation which is anti to at C and syn at G? So what is this anti and syn? The C1 of pentose sugar in the nucleotide is linked to the N1 or N9 position of pyrimidine or a purine base through glycosidic bonds. The syn and anti conformation are two conformational forms of the nucleotide around this bond, and the two forms are interconvertible also, that is by you can rotate the glycosidic by rotating the glycosidic bond without the need to break the bond. So which type of DNA has glycosyl bond conformation which is anti at C and syn at G is zDNA? Answer is option 3. Then next one. One of the first ribozymes to be discovered was RNase P, which is involved in. So RNase P, it is involved in generating tRNA molecules from larger precursor RNAs. Answer is option 1. Only one round of DNA replication occurs during each cell cycle in eukaryotes because helicase loading and helicase activation are highly regulated to occur only during G1 and S phase respectively. Okay. So loading of helicase, what will happen when once the loading of helicase happens, there is unwinding of long stretches of DNA. And once this replicative helix is activated, that is helix activation, the origin of DNA is unwound and bidirectional replication forks are established. That is called as loading of helicase loading and helicase activation. So only one round of DNA replication occurs during each cell cycle in eukaryotes because helicase loading and helicase activation are highly regulated to occur only during the G1S phase respectively, because that is at that phase only, that is at the S phase only DNA replication occurs. So answer is option 1. Then, how many subunits does the histone chaperone CAF1 has? So, chromatin assembly complex 1, that is called as CAF1. It is a major histone chaperone involved in the deposition of histone H3 and H4 into the nucleosome. And CAF1, it has got 3 subunits. So, the answer here is option 3. Then, during transcription, the DNA nucleotide encoding the beginning of the RNA chain is designated as. So the site on the DNA from which the first RNA nucleotide is transcribed, it is called as the +1 site. Answer is option 3. Then next one. The large subunit of Pol II has carboxy terminal domain which contain a series of repeats of the heptapeptide sequence. The number of these repeat in yeast, Drosophila and humans. It is 27, 45 and 52 respectively. Option 1. Next one. RNA polymerase 1 transcribe which gene? It transcribes rRNA. Option 2. Viroids are parasites of. So viroids are obligate parasites of cells transcriptional machinery. So the answer here is option 2, that is host transcription machinery. The origin of satellite viruses is most likely from. So the origin of satellite virus must be a relic of the RNA world. Answer is option 4. Then rotavirus encode 12 polypeptides out of which dash are located in the virion and dash form the inner shell, form the middle shell and dash form the outer shell. So rotavirus encode 12 polypeptides out of which 6 are located in the virion and 3 form the inner shell, 1 form the middle shell and 2 form the outer shell. So the answer is option 1. In a virus infection experiment in cell culture, at multiplicity of infection, MOI of 1, what percentage of cells will be infected? So the probability of cells that will be infected is given by P is equal to 1 minus E raised to minus MOI. So 1 minus E raised to minus 1, if you substitute, you will get it as approximately 0.63 or 63%. And as per the options, the answer which is more near to 63 is about 67%. So answer is option 3. The next one. Which of these viruses is reverse transcribing virus? So it is option 2, that is cauliflower mosaic virus. Answer is option 2. Then which one of these pattern recognition receptors does not recognize double-stranded RNA during virus infection? So AIM2, it recognizes double-stranded DNA, not double-stranded RNA. Answer is option 4. Then which of these viruses undergo latency in host cell that involves integration of viral genome into host genome? So it is adeno-associated virus. Answer is option 3. Then the size of the repeat sequence in VNTR is generally how many base pairs? So VNTR is a variable number of tandem repeats and these are short nucleotide sequences. They vary in copy number and the number of times that particular sequence is repeated. So the answer here is 2 to 100. Then next one. Streptococcus pyogenes is the cause of streptococcal pharyngitis, which is better known as strep throat. Answer is option 1. Then Mycobacterium leprae, a relative of Mycobacterium tuberculosis, causes the disease leprosy, which is formerly known as, it was called as Hansen's disease. Answer is option 1. Then, this is gram-positive, highly motile, spiral-shaped bacterium which is associated with gastritis and ulcers and colonizes the non-acid secreting mucosa of stomach and upper intestinal tract. Identify the bacterium. So it is called as Helicobacterium pylori or H. pylori. Answer is option 3. Then next one. Rickettsias are divided into 3 groups based on the clinical disease they cause. Which of the following is not one of the groups? So it is plasmosis group. All the others are belonging to the group, whereas plasmosis group doesn't belong. Then next one. Choose the incorrect statement about microscopy. Magnification can be increased without limit but resolution cannot. Okay. Limit of resolution of a light microscope is 2 micron meter. Then magnification of a compound microscope is the product of the magnification of its objective and ocular lens. Resolving power is directly proportional to the wavelength of illumination used. Resolving power is inversely proportional to the wavelength of illumination used, and limit of resolution of light microscope is 2 micron meter. These are the incorrect statements. So in the answer key it is given only 2 as the answer, but as per me 4 is also an answer. Then, which of the following is a pair of omega-6 and omega-3 fatty acid? So answer is linoleic acid and linolenic acid. Answer is option 3. Then which of the following groups of archaea has mesophiles? So it is Thaumarchiota, Archaeota. Answer is option 2. Then next one. Choose the correct statements among the following about electron transport. So carbon monoxide binds cytochrome A3 and thus preventing oxygen reduction. Yes. Azide binds tightly to NADH dehydrogenase and inhibits flow of electrons. Azide inhibits oxidative phosphorylation via inhibition of cytochrome oxidase. So option B is not correct. Then uncouplers inhibit ATP synthesis without affecting ATPase. That is also correct. Then next one. Dinitrophenol and dicumarol are lipid insoluble uncouplers. They are lipid soluble. So option D is also not correct. So answer is A and C only, option 1. Then next one. Match the groups listed in column A with their source of energy and carbon from those listed in column B. So chemolithotroph, they are inorganic compound and carbon dioxide. Then next one. Chemoorganotroph, the glucose both as source of energy and carbon. Photoheterotroph, photoheterotroph is naturally light and heterotroph is organic compound. Photoautotroph, light and carbon dioxide. So answer is option 1, A2B1C4D3. Then next one. Chlamydia they are obligate parasites and causative agent of trachoma in humans. First option itself is correct. Then next one. Which is not true about both Equifex and Thermotoga? Both belong to domain Archaea. It is not Archaea, they belong to domain Bacteria. So first option itself is the right answer. Then What is not true for shock sensitive transport systems? So what is this shock sensitive? So when bacterial cell it is suspended in hypertonic solution, what will happen? It will lose water and the cytoplasm shrinks. Now if you collect all these cells in the hypertonic solution and you resuspend them in water, the cytoplasm will rapidly expand and active trans expand and the cell will regain its size. Okay. Now active transport system remains relatively intact after this treatment and are classified and they are called as osmotic shock resistant. Those that have lost this active transport system, they are called as osmotic shock sensitive. So once you take a bacterial cell from hypertonic solution and resuspend in water, some cells their active transport system remain relatively intact and they are called as osmotic shock resistant, whereas those which have lost the active transport system are called as osmotic shock sensitive. So what is that they have asked? What is not true for shock sensitive transport system? So they are not functional in cells subjected to osmotic shock. Yes, that is correct. They have a periplasmic binding protein. Okay. Now one salient feature about the shock sensitive active transport system is that they require a periplasmic binding protein that tightly and specifically binds the molecule to be translocated. So that is true in case of shock sensitive transport. They are energy independent. That is not the correct answer. That is not true with regards to shock sensitive transport system. So answer is option 3. Now next one. Which of the following features is not true for archaeal cell walls? Archaeal cell wall are made up of either pseudomurine or protein, the S layer. That is correct. The walls are not susceptible to lysozyme hydrolysis. Yes, the sugars in archaeal cell walls are linked by beta-beta 4 linkage. They are linked by beta 1,3 linkages. So option 3 is not correct and that is your answer. Then which channels maintain cell integrity in dilute solutions? So it is mechanosensitive channels. Answer is option 2. Then what is not true for domain archaea? So multiple origin of replication in Sulfolobus species. That's correct. Archaea are thermophiles. They are not only thermophiles, even halophiles are also present among archaea. So option 2 is not correct. Then chlorosomes lie immediately underneath the cytoplasmic membrane of which group of photosynthetic bacteria? So they are found in and chlorosomes they are photosynthetic light harvesting complexes and they are found immediately underneath the cytoplasmic membrane in the case of green sulfur bacteria. Answer is option 1. Then next one. Which of the following is not an example of multi-enzyme complex? Pyruvate dehydrogenase, yes. Pyruvate carboxylase, no. So answer is option 2. Then reverse electron transport is observed among. So it is observed among chemolithotrophs. Answer is option 3. Then next one. Which of the following statements are true for bacterial electron transport? So E. coli transfer electrons from reduced quinones to cytochrome O, which is the major one when oxygen levels are high. And okay, that's correct. Then important difference between electron transport chain in bacteria and that in mitochondria is that the former is branched. Yes. Cytochrome D is an alternate to cytochrome O in bacteria to rapidly reduce oxygen during high concentration. No. Then menaquinone is important for electron transport in anaerobic gram-negative bacteria. So D is also correct. So answer is option A, B, D, only option 4. Then next one. Enter Duodorf pathway has one of the following unique enzymes. So the entrant Duodorf pathway, it is an alternate series of reaction that catalyze glucose to, not catalyze, catabolize glucose to pyruvate, and it uses different set of enzymes different from those used during glycolysis and pentose phosphate pathway. Then it uses 6-phosphogluconate dehydratase and 2-keto 3-deoxyphosphogluconate aldolase to create pyruvate from glucose. So the answer here is 6-phosphogluconate dehydratase or dehydrogenase. Answer is option 2. Then oxaloacetate is replenished during TCA cycle by which of the following enzyme activities? So it is pyruvate carboxylase. It catalyzes conversion of pyruvate to oxaloacetic acid when abundant acetyl-CoA is available and thereby replenishing the Krebs cycle. The next one. An enzyme was treated by an inhibitor and it was observed that inhibition could be overcome at high substrate concentration. The larger the inhibitor concentration, the larger was observed Km value. Based on these observations, identify the type of enzyme inhibition. So here the Km value is large, then you can overcome the inhibition by adding more substrate, and the Vmax is the same. So it is naturally a competitive inhibition. Answer is option 2. Then microbial activity is measured by dehydrogenase assay using one of the following reagent. So which is the reagent? So it is dehydrogenase activity can be measured using different tetrazolium salts, that is 2,3,5 triphenyl tetrazolium chloride. So answer is option 2. Next one. Bacterial endospores have large amount of dipicolinic acid, and that's a direct question. Answer is option 3. Homofermenter and heterofermenter lactic acid bacteria are differentiated based on fermentation products they form. This difference is because of the enzyme activities present in them. Choose the correct statement. So homofermentive bacteria produce only lactic acid, whereas the primary product during glucose fermentation, while heterofermentative bacteria produce ethanol or acetic acid or even carbon dioxide other than lactic acid as byproducts during glucose fermentation. So EMP pathway is used by homofermentative type and it uses the enzyme aldolase or fructose 1,6-diphosphatase, and heterofermentative bacteria use phosphoketolase pathway and they lack the aldolase enzyme. So the answer here is okay. Homofermenters lack transketolase but have aldolase. Heterofermenters have phosphoketolase and lack aldolase enzyme. That is more correct. Answer is option 2. That is heterofermenters have phosphoketolase and lack aldolase enzyme. The next one. An organism lacks cell wall and yields no reaction with gram stain. Cells are small coccoids or highly pleomorphic and require sterol for their growth. Identify the organism. It is mycoplasma. Answer is 2. Which of the following is not a halophile? Halobacterium, Natronococcus, Halococcus, Heliobacterium. Heliobacterium is not a halophile. Then which pair of bacteria are part of the group of electricity generating bacteria? They are Geobacter and Schwannella. Answer is option 4. Then a student could not distinguish correctly between Salmonella and Shigella based on sugar fermentation test, although E. coli was distinguishable. What might be the mistake done by the candidate while preparing the triple sugar agar? So triple sugar agar is designed to differentiate among organisms based on differences in carbohydrate fermentation pattern and hydrogen sulfide production. And carbohydrate fermentation, it is indicated by the production of a gas and change in the color of the pH indicator from red to yellow. So now Salmonella gives red slant yellow bud. Shigella also gives red slant yellow bud, but E. coli gives yellow slant yellow bud. Now red slant yellow bud, it indicates dextrose fermentation, and yellow slant yellow bud indicate fermentation of dextrose, lactose and or sucrose. So now I told you that Salmonella gives red slant yellow bud. Similarly, Shigella also give red slant and yellow bud. It indicates dextrose fermentation. So you cannot say that they forgot to add lactose or they did not add sucrose. And option 4 is not told in the experiment. So it is not the carbohydrate source that is absent. It is option D, that is they did not add iron. Why? Because if they had added iron, then H2S production could be distinguished between two bacteria, as H2S is produced by Salmonella and Shigella but not produced H2S is produced by Salmonella and Shigella does not produce H2S. E. coli also, it does not produce H2S. So adding or not adding iron did not hinder with E. coli detection. But E. coli could be distinguished based on yellow slant and yellow bud. But Salmonella and Shigella, they give red slant and yellow bud. But if you had added iron, then H2S production could be distinguished between the two bacteria, as H2S is produced by Salmonella and Shigella does not produce H2S. So they had forgot to add iron. That is why they could not distinguish correctly between Salmonella and Shigella. Then next one. Given below are two statements. One is labeled as assertion and the other is labeled as reason. Assertion. A mutation in the LAC-Y gene results in the bacterium failing to grow on medium where lactose is the sole carbon source. The reason. The LAC-Y gene encodes for beta-galactosidase, the enzyme that breaks down lactose. In the light of the above statements, choose the appropriate answer from the options. So LAC-Y, it codes for permease that helps to transfer lactose into the cell. So what they have given, a mutation in LAC-Y gene results in bacterium failing to grow on medium where lactose is the sole carbon source. Of course, because LAC-Y codes for permease that help to transfer lactose into the cell. Now when the LAC-Y itself is mutated, lactose will not be transferred into the cell and it will fail to grow on lactose medium when lactose is the sole carbon source. Then next one, reason is the LAC-Y gene encodes for beta-galactosidase. It is not beta-galactosidase, it is permease that lactose-Y encodes. So the answer here is A is correct but R is not correct. Answer is option 3. Then next one. The following is the list of steps involved in the transfer of bacterial DNA by generalized transduction. Which option represent the correct sequence of steps? So first one will be injection of P1 phage DNA into bacterial host. Then comes fragmentation of host genomic DNA by phage encoded endonuclease. Then come packaging of the host genomic DNA fragment into P1 phage head. Then comes release of the transducing particle from the bacterial host cell. Then injection of DNA of transducing particle into bacterial host. So this is the order and it is seen in answer 3. Option 3 is the answer. Then, which of the following statements made with regard to plasmid incompatibility are true? Plasmids having the same origin of replication belong to the same incompatibility group. That's correct. Plasmids having different origin of replication belong to, that is not correct. Plasmids of the same incompatibility group are compatible. No, they are plasmids of the same incompatibility group are incompatible. So D is correct. So answer is option 2, that is A and D only. Then choose the most suitable option correctly matching the DNA repair system listed in column A with the kind of DNA damage from among those listed in column B. Photoreactivation, it is cyclobutane ring. Mismatch repair is replication errors. Base excision repair is oxo-G, and NHEJ repair is double stranded break. So answer here is A2, B4, C3, D1. Answer is option 1. Then given below are two statements. One is labeled as assertion and the other is labeled as reason. Assertion. Mutation in the codon 5' ACG 3' to ACC 3' is a silent mutation. The new codon created by the mutation encodes for the same amino acid as the original codon. Yes. Now if you see this, that is mutation in the codon ACG. So the G is replaced by C. That is G at the third position is replaced by C, but it is a silent mutation because it codes for the same amino acid. So both A and R are correct, and R is the correct explanation of A. Which of the following phage exhibit non-integrative lysogeny? It is P1. Answer is option 4. Which of the following is a Drosophila transposon? P. That is a Drosophila transposon. Mapping bacterial genomes by interrupted mating uses. So it uses HFR strains. Answer is option 2. Then R plasmids are medically important because they carry a large number of antibiotic resistance cassettes. That itself is correct answer. Option 1. Which of these cannot be the effect of transposition? Activates gene expression? Yes. Inactivates gene expression? Yes. Confers on the host bacterium the ability to act as donor in conjugation. That is not the effect of transposition. It is the presence of an F plasmid where the donor will, where the bacterium will act as the donor in conjugation. So answer is option 3. Then the following is a list of steps involved in cloning a gene. Which option represent the correct sequence of test? So the first one is amplification of the gene by PCR using Taq DNA polymerase. Then comes ligation of gene into TA cloning vector. Then comes transformation with blue and white selection. Then comes isolation of plasmid DNA, and then comes the RFLP analysis. So the answer here is 5, 4, 1, 3 and 2. So answer is option 4. Then choose the most suitable option matching the enzyme listed in column A with their application from among those listed in column B. Alkaline phosphatase. Alkaline phosphatase is dephosphorylation. DNA ligase is for gene cloning. Type 2 restriction enzyme is for RFLP analysis. And DNase 1 is for the DNA footprinting. So the answer is A2, B4, C1, D3. Option 1. Given below are two statements. One is labeled as assertion A and the other is labeled as reason R. Assertion A. Some restriction enzyme exhibits star activity. Okay, now what is star activity? That is seen under non-standard condition in case of restriction enzyme, because at those conditions, that is these non-standard condition, they will be capable of cleaving sequences which are similar but not identical to the defined recognition sequence. This activity is called as star activity. Okay. And the reason, the same sequence of DNA can be recognized by two different restriction enzymes. That is also correct, but it is not the correct explanation of A. So both A and R are correct, but R is not the explanation of A. Answer is option 2. Then which of the following statements made with regard to Sanger's method based automated DNA sequencing are true? Uses DNA polymerase 1. It does not use. Sanger's method. Okay, it does not use DNA polymerase 1. Uses chain termination to detect DNA sequence. Yes. Uses PCR to carry out sequencing reaction. No. 1 minute. Yes, it uses PCR for carrying out sequencing reaction. Yes. Then uses fluoro tagged deoxynucleotides. It uses dideoxynucleotides. So D is also not correct. So answer is B and C only. Option 4 is correct. Then choose the most suitable option matching the method listed in column A with its application from among those listed in column B. Northern blotting is for RNA analysis, Western blotting is for protein analysis, phage display is for epitope mapping, and electroporation is for DNA delivery. Very easy. So the answer is A2, B4, C1, D3. Then which of the following method is most suitable for screening cDNA expression libraries? It is immunological screening. That is the most suitable method for screening cDNA libraries. Then when making radio-labeled probe using Clino, which of the following methods would be used? Random priming. Now random priming, it uses short nucleotides, that is around approximately 6 nucleotides long, and these are random sequences which prime the denatured target DNA at numerous sites. Then what will happen is the Clino fragment of DNA polymerase 1 is then used to synthesize new strands of DNA from these primed sites. And if you add a radioactive nucleotide, it will result in labeled probe which will be suitable for southern hybridization. So the answer here is option 2, that is random priming. The next one. The use of YARCs in whole genome sequencing is associated with certain problems. Which of the following is not one of them? Occurrence of chimeric inserts, yes. Frequent point mutation arising with time, no. Sorry. Answer is option 2. The next one. Which of the following cannot be carried out using electrophoresis through agar? So determination of molecular weight of DNA, yes. Purification of DNA fragments, yes. Electrophoretic mobility shift assay and DNA footprinting. Okay. So electrophoretic mobility shift assay, it is based on the protein DNA interaction. It is based on observation that protein DNA complexes migrate more slowly than free linear DNA fragments when subjected to non-denaturing polyacrylamide or agarose gel. Whereas DNA footprinting, it uses page gels to study DNA protein interactions. So answer here is option 4. It cannot be carried out using agarose gel electrophoresis. Then which of the following is not a promoter that drives recombinant protein expression in E. coli? It is CAMB. It is used as a plant transformation promoter. Answer is option 3. Then choose the most suitable option matching the nuclear component listed in column A with its function from among those listed in column B. Nucleolus. Nucleolus is ribosome assembly. Nucleoplasm suspension medium for nuclear organelles, nuclear lamina structural support, and nuclear pore complex is movement of RNA to cytoplasm. Answer is option 4. A3, B2, C4, D1. Then given below are two statements. One is labeled as assertion A and the other is labeled as reason R. The addition of amino acid motif KDEL to the carboxy terminus of a protein that is normally secreted will block its secretion. Next, reason. Proteins bearing KDL motif bind to specific recycling receptors in the membrane of the endoplasmic reticulum Golgi intermediate compartment and are selectively transported back to ER. Yes, because KDEL motif is for directed to proteins will be directed back to endoplasmic reticulum. So in the light of these above statements, choose the most appropriate answer from the options given below. Both A and R are correct and R is the correct explanation of A. Which of the following statements made with regards to membrane transport are true? Channel proteins form open pores through which suitably sized molecules ions can cross the membrane. That's correct. Carrier proteins selectively bind specific molecules and transport them across the membrane by undergoing a conformation change. Yes. Transport of molecule across membrane through channel proteins and carrier proteins always involves active transport. So carrier proteins need ATP to change conformation, whereas channel proteins are not used in active transport because substances can move only through them along a concentration gradient. So option C is not correct. Small uncharged molecules can diffuse freely through the phospholipid bilayer. That is also correct. So answer is option 3, A, B and D only. In mitosis, what is the correct sequence of steps from those listed below? Chromosomes are aligned at the equator midway between the spindle poles. Chromosomes condense, yes. Option that will be the first one. Then comes breakdown of nuclear envelope. Then comes chromosomes are aligned at the equator midway between the spindle poles. Then daughter chromosomes move to the wait. Yes. Sister chromatids synchronously separate to form two daughter chromosomes. Yes. And then the daughter chromosomes move to the poles of the spindle. Answer is option 1. That is 2, 5, 1, 4 and 3. Then proteins that transport peptides or small molecule across membranes using the energy of ATP hydrolysis are. They are called as ABC transporters. Answer is option 1. Actinobacteria are common producer of which of the following compounds? So it is antibiotics. From 76 to 79 it is fermentation technology and I am not good with the subject so I have not answered that. Okay. Then next one. Which of the following microorganism is generally used as the test organism during steam sterilization process? It is Bacillus stearothermophilus. Answer is option 3. For the long-term storage, which of the following strategies is best suited for the higher viability of microbial cultures? So they are stored at minus 80 degrees Celsius. That is for long-term storage. Then, in the case of recombinant therapeutics, which of the following factors is not detrimental to its final market cost of the product? So it is product labeling and branding. Answer is option 4. To recover an intracellular product from a bacterial culture, which of the following enzyme is predominantly used for cell wall disruption? It is lysozyme that is used. Answer is option 3. Then gluconobacter is used in the preparation of vinegar. Mare milk is traditionally used to make which of the following fermented product? It is cow maize. Then the flavor enhancement and eye formation in Swiss cheese are done by which of the following microorganism? It is Propionibacterium fruderensi. Answer is option 2. Lactobacillus bulgaricus and Streptococcus thermophilus are generally used to prepare which of the following fermented dairy products? It is yogurt. Answer is 2. The concept of appertization is used for the preservation of which of the following items? So appertization refers to the process where the only organisms that survive processing are non-pathogenic and are incapable of developing within the product under normal conditions of storage. So appertized products have long shelf life even when stored at ambient temperatures. So it is used for the preservation of canned food products. Answer is option 4. Oxidation of organic acid in foods lead to the formation of which of the following ions? So it is carbonate ions. Answer is 2. Pseudomonas fluorescens is primarily responsible for the green rot of eggs. 1. The book for all households or the art of preserving animal and vegetable substances for many years was authored by. It was by Nicholas Appert. Sugarcane's red rot is caused by which of the following bacterium? It is Colletotrichum facultum. The next one. Harold Henry Flor proposed which of the following hypothesis? Gene for gene hypothesis. Answer is option 1. The host non-specific TAB toxin is a dipeptide. Answer is option 1. The MacArthur Forest hydrometallurgical process is used for the recovery of which of the following metals from their natural ores? It is gold. Answer is option 1. The term metagenomics was coined by which of the following scientists? Joe Emil Handelsman. Answer is option 1. The phenomenon of magnetotaxis in certain bacteria is due to the presence of magnetite particles. The salt tolerance in Ectothyroidospira halochloris is conferred by. It is conferred by ectoins. Answer is option 2. Which of the following is not an international culture collection center for their long-term storage? It is American type culture collection. So that's it. Thank you for watching my channel. Please do like, share and subscribe. Thank you.